lead.
Hey there, everybody. Welcome. We apologize if you had trouble getting on this morning to the Zoom. I did too. It's a little bit panicky here. Hopefully we've got everybody on that needed to. I know we had 2 classrooms that were logging on today, so we are very happy to have you here. And if this is your first time on a visit to Archbold, whether it's a virtual visit or a in-person visit, I am so glad that you're here to share a beautiful morning together. If you were on last week, you might remember that we were looking at some animal tracks. I brought out some plaster molds and was sticking them in the sand, and so you could see what some of the animal tracks look like. Well, the reason I'm sitting on the ground right here is I've got a perfect track, and I'm going to try to get real low here, and hopefully you can see this too. Right in front of me here. This is not a deep track, but can anybody notice there's 2 paw prints here, 1 paw print here, and then another small paw print right here. And if you know what it is, or you've got a guess, stick it in the chat there. I would love to see some guesses. This is a really distinct animal track, and a lot of the times when I see it, I know what it is, even though I can't actually make out the little paw prints, because I always see this shape here. It looks kind of like a heart. So that gives it away. This is a rabbit, a cotton-tailed rabbit track. A lot of times when you see them, that you won't really see the exact little paw prints there, but you'll still see that shape. So I thought that'd be fun to start off with. Well, anyway, so welcome back to Archbold. Turn this up a little bit. And the way this works is we take about a half an hour to look around and see what we can find out here. I've got my selfie stick. And then we always have special guests, and we do a question and answer at the end. Today, our special guest is one of the research interns in the plant ecology lab, AJ Jackson. And we're going to do something a little bit different today. So instead of showing video clips on things, she's going to come in and tell us what it's like to be an intern here at Archbold, and even use her cell phone to show us what the plant ecology lab looks like, what the offices look like. So that'll come up a little bit later. So that should be pretty cool. Margaret, can you share your screen and show us the picture of Florida from outer space, please? We are at a really interesting place. Archbold Biological Station is located on one of the higher parts of Florida. And you can actually see from outer space, I circled it here, there's this strip of white sand in the middle of the state. And you might think, what could that be? Florida doesn't have any mountains. It's generally pretty flat, pretty low, close to the ocean. But in the middle here is a big pile of sand. If you imagine the ocean higher than it is today, imagine sand being pushed to the middle of the state and forming an old sand island. That is where we are. And because it's a big, tall, high and dry spot, that means that the habitat here has been here much, much longer than all of those other habitats around it, because they were covered by the sea if you go back around 1 million years ago, and this was an island. So this is a very ancient habitat, and if you think about an island like the Hawaiian Islands or the Galapagos Islands, you might think, ooh, they've got stuff there that you don't find anywhere else. That's the same right here. This isn't the only island. It's called the Lake Wales Ridge. There's other sand ridges in Florida too, and on them you find this very special habitat, the Florida scrub. That's what we're talking about today. All right. Oh, and you see a little logo there for Archbold on the south end. Boop, that's where Archbold is. Up at the north end right there, just to the right of it, to the east, that's Orlando, if you've ever been to Disney World before. All right. You can stop sharing. Thank you. And what I'd like to do today is something a little bit different than I've done before, because I'm going to give you an assignment here. See, I've got next to me this tripod. I'm going to put my camera on the tripod and give you a look here. And what I want is for you to practice your scientist skills. Now, when you're learning about science in school, you'll learn about the scientific method. And you'll hear about making a hypothesis, which is like, sometimes people call it an educated guess, but that I don't really like that. It's more like a proposal or a proposition on what you think is going to happen in nature based on some knowledge you already have. So you have a hypothesis, and then you need your experiment. You have methods and procedures. Then you have results and conclusions. Well, in the real world of science, sometimes it works like that, but it could also look differently. Maybe it's not an experiment. Maybe what you're doing as a scientist is making an observation of something people haven't seen before. Like, say you see that a bird is eating something that nobody knows it eats that, and you're telling people about that. Or maybe you're helping to make a model of something, trying to understand how something works. That can be science too. All of those things have in common observations. So that's what your task is going to be today, is to practice making observations. So let's see. I'm going to take my camera off my selfie stick and put it up here. Sorry, I got to cover this for just a second here. You can see my beautiful hand. Put it down on here. And I'm going to switch it over. There we go. Beautiful. A little lopsided, but see if I can even it out a little bit. There we go. Nice. Nice. Okay. So, hey, there I am. There's my selfie stick. Okay, so behind me here, we've got some scrub at Archbold. And what I want you to do is have a partner, have somebody that's kind of close to you, or if you don't have anybody else watching with you, you can just say this out loud or type it in the chat. But what we're going to practice first is I notice statements. These are observations. And I want you to think like you've never seen any of this before, and you're not drawing conclusions. So an observation would be, I notice that there's clouds in the sky. You wouldn't say why there were clouds there or what clouds are made out of. It's just that you notice there's clouds there. That would be an observation. So I'm going to go back around the other end there so I can read the chat. Would love to see some observations in the chat. So here we go. I'll give you about 30 seconds. Let's see some observations. And remember, if you have people with you, you can make I notice statements right to them. If you're in a classroom, just shout it out, your I notice statements. I see we've noticing trees, lots of sand, lots of sand. Yeah, it's a cloudy day. Oh, it was very misty, foggy this morning when I came out here a little earlier. And I'd say that when you're describing the scrub, sand is the #1 thing I would say. And palmettos, yes, we've definitely got palmettos here. If you don't know what a palmetto is, here's a palmetto. That palmetto is a saw palmetto. You gotta be careful because you can cut your fingers on it. Very good, very good. Okay, the next part here is we are going to do I wonder statements. So first, we're just focusing on what we describe. We're using our eyes. If you were out here, though, you could touch, you could smell, and you could add those to your I notice statements. Now, I wonder. Remember, think like a scientist. If a scientist is going to come up with a hypothesis, but how they think things work out here, they've got to start with those observations and now start asking questions. So let's see some questions. What do you wonder? Imagine you're a scientist, and if you come up with an I wonder, yell it out loud or type it in there. Wonder what kind of tree that is. Yeah, our Riley family in Virginia. What other kinds of questions do you have? Why is there so much sand? I wonder why plants grow in certain places, but not others. We notice, yeah, different plants in different spots. I see a clear part of the sky that looks like a boot. That would be an I notice statement, not an I wonder. I wonder would be, I wonder how it got that shape. Okay, we'll be like, how come clouds don't just cover the sky all evenly? How come there's clumps of clouds? That'd be a good I wonder. Awesome. Nice job. And last part here is it reminds me of. This is where we're going to take the observations and the questions and start making connections between them. So say you wondered, what kind of tree is that? Well, now, what does it remind you of? Or maybe you noticed that certain plants grow in certain spots. What does that remind you of? Same thing, if you're in a classroom, shout it out, shout it out. And you can also type it here. Reminds me of the beach. Yes, perfect. Connie, I love that one because this used to be a beach. We could go back in our time machine. This was a beach. Can you imagine just a couple miles from here would have been manatees and dolphins swimming around? Anybody else? What do things remind you of? Why haven't the bushes grown into the sand? Okay, cool. Now my idea here is not to answer all of these questions, though some of them we can get to, and we've got that question and answer at the end. But this is an activity you can do anywhere. The next time you go outside, you could be looking at anything. You could be in a forest or a park. But you could also just be looking at the sidewalk and you see some plants growing up in the sidewalk. Or maybe it's not plants you're looking at. Maybe it's snow or ice, depending on where you are. And try that again. Describe what you see, only descriptions, and then start asking questions and making connections. So what do we see out here? Someone noticed the trees. These tall trees here, those are all slash pines. Slash pines. But the dead tree on this side behind me, that one is a sand pine. They're the two pines that we have out here in the scrub. For people who know their pines, there's also longleaf pines in this area, but we don't generally see them in the scrub habitat. Nice job. Okay, I've come back around. Switch back here. Oh, come on. There I am. I'm sweating out here. That was really fun. I hope you liked that too. So without further ado, I'd like to bring on our guest. AJ is going to come on twice. This first time, she's going to tell you a little bit about what it is that she does here. While she is on, I'll turn my camera off because I'm going to walk out. There's another spot I want to get to, so I'm going to go for a walk out there. So AJ, are you here?
I am present. How are you, Dustin?
Hey, good to have you on. I'm so glad you're here. I love those photos. Okay, I'm going to turn my screen off. You've got the next 5 minutes.
Okay. Got you. Hello, everybody. I hope you guys are doing great. So my name is AJ. I am an intern here at Archbold. I'm a Bond Jordan Ecology program intern here at Archbold. So to give you some background about me, I'm in my post-baccalaureate internship. I graduated from Virginia Tech in May of 2020, and I'm going into my master's program in this fall. And I've really enjoyed my time here at Archbold. I really enjoy the people here. I love what I do, which I'll be talking about in a little bit, but I work in the plant lab, as they said, multiple times. And we typically have 2 to 3 interns, and we come from everywhere. We all have different backgrounds. To give you a little idea, some people come from entomology backgrounds. I came from a wetland biogeochemistry background, so very diverse. Can you click next for me, Margaret, so I can talk about what we do in the plant lab? So I thought of the top 4 things that we do. And I think the most important thing is we do a lot of field work, demography, which is just looking at what's there, what's alive, getting measurements. We take clippings, we do introductions of plants. And with field work, that means we do a lot of data collection and analysis. We have a herbarium, which I'll be showing you later on. We do a lot of upkeep of that. And then every intern has an intern independent project. So right now I'm kind of still going through mine, but hopefully the next time I talk to you guys, I'll be telling you guys what I learned in my internship project. And then this next slide is some pictures of our field days, which can be really long, but they're so amazing. I'm gonna start with one of my favorite projects, which is Florida Zizyphus. Can everybody say Zizyphus for me? It's one of my favorite words now. And we did demography and sampling the whole month of January. It was my first project here at Archbold. And Zizyphus is just a really interesting plant. It was, endangered for a really long time. They thought it was extinct until like 1987. It was rediscovered. And then in 2001, like 8 more populations were discovered. And then that's when a lot of places like Archbold and some universities took on projects to then like keep Zizyphus alive. So since those projects have started, they've planted like 600 plotted plants and about 6,000 seeds. That's a lot. So just to keep you in mind of how important Zizyphus is and how important it is to keep rare endangered species of plants in this area in Lake Wales Ridge alive. In that bottom left, you can see we're doing some collections of Diceranda funiculata. And that's a rare mint species here in Florida. It smells so good. And then on the bottom right, that is us out there. It was really cold that morning, contrary to believe. It gets kind of cold here in Florida. And we were doing Florida rosemary demography. So as you can see to the far right, that is our RA, Lexi, with her retractable measuring Florida rosemary, which can get really big. And we were looking at the sex, whether it was male or female or not. And yes, it was a great project. I enjoyed that so much. So those are my favorite 3 projects so far that we've done. But like I said, I'll be here for a while longer. We have so many more things to do. So hopefully I can tell you guys about that later as well.
Awesome. Thank you, AJ. I've, while she was talking, I've been hiking around out here because I wanted to find this other plant that's pretty cool. And I should say with all our COVID restrictions, I actually haven't gotten to know the interns really this year. It's been really a bummer. I only got to meet AJ last week when we met to talk about this program. So that was new information for me too. That was pretty cool. I do want to give a book recommendation. Florida Wildflowers. This is my favorite plant guide for the flowers here at Archbold and in Florida. There's been multiple versions of this over the years, written by Walter Kingsley Taylor. Love it. Okay. So we've got this cool plant right here, and I'm gonna get down low, take a look, and then we'll look at it with the macro lens in a second too. Take a look at this little guy, and you'll notice that I'm not touching it. Oh, I'm not touching it. I'm just looking at it, and there's a reason for that. Does anybody have any idea what this little white flower could be? And I'll say there's about probably a dozen of these I've seen on this trail right here, and they're really pretty. They love sandy roads like this. The reason I'm not touching them is because the name of this is finger rot. Finger rot, also known as spurge nettle. And if the name finger rot sounds like maybe you shouldn't touch it because your finger will rot, you are right. There are little hairs on here. I see someone just noticed that. Those little hairs have chemicals on them. This plant will sting you. It has its own defense. All those little hairs on there have chemicals that some people are very allergic to. Some people not so much. I'm not going to test that out. But if you go online and start reading stories about people getting stung by nettles, oh my goodness. I mean, it can kill certain people. Not everybody, but if you're really allergic to it. For other people, it might burn for an hour. something like that. Yeah, there's a bunch of little ants. Thing is, once you get close up to the ground, there's always ants, and on trees. People don't realize that, but they're all over everything. They're checking everything out. Okay, I see the leaves. I see the little flowers. I see something in the middle there. I guess maybe that's a bud for another flower that's going to open. That's what it looks like. But let's switch around our macro lens and take a closer look at this. Okay, I'm flipping it around. Be blurry for a second till I get it close up here. Here we go. Oh, there we are. Oh, really pretty little flower. Get it. Oh, there it is. There it is. Oh yeah, gorgeous little white flower. Let's see some of these hairs. Oh yeah. They do not look too inviting. Does it grow bigger than that? No, I've never seen them bigger than this. I mean, maybe they get like a couple inches, but they're always really small like this. And when we think of plants, we don't always give them enough credit, right? We don't really give them enough credit. We just think of, they're just kind of sitting there. But plants have to think about defending themselves too. So they do that in different ways. Some of them have spikes. These spikes here are not like a cactus. The spike itself isn't going to hurt you. It's the chemicals on the spikes that will hurt you. There's the leaf there. See all those bits of sand. Remember, we're on the Lake Wales Ridge, so we've got that. People call it sugar sand. Oh, nice. Nice. All right, I'm going to... Oh, actually, I wonder if I can get one of these little ants. They're moving around, but let's give it a try. Oh, it's not easy. I'll try once, right? Oh, there he is. Okay. There are a bunch of little ants running around here, and I can see some of them are carrying things. Yeah, the sugar sand does look like sugar. And if you think about it, when you're potting plants at home, you don't want to, generally, depending on what it is, but generally you're not, say you want to grow tomatoes or something, you're not going to stick them in a pot of white sand. You want to have organic material in there. You want nutrients for them. So the scrub does not have a lot of nutrients in the sand. The plants here have to be adapted for it. It's a little bit like a desert. Okay, there's another flower I want to show you. And it's just over here. Let me get my Wi-Fi here. And where did it go? Oh, there it is. It's still winter, but spring is in the air. Spring is in the air, and I was so happy when I came out this morning and saw that some of the flowers are starting to bloom. Here we go, right here. Pretty little flowers. Feel free to take a guess in the chat. These flowers look like little bells. They hang down. Here we go. These are called shiny blueberries. Yeah. No, these ones are not Lyonia, though they look a little bit like shiny Lyonia. Let's flip this around. Put the macro lens on again. Let's take a look. There we go. Beautiful. Yeah, these have just bloomed. Just bloomed. And these will get tasty little blueberries. They're not real big, but they are very tasty. I usually see the blueberries like the first week of June. And let's look at another one. See, some of these flowers haven't even opened up yet. Just about to open up. We have 3 different kinds of blueberries here at Archbold. 2 of them are little short ones like this called low bush blueberries. And then one of them is the regular high bush blueberries, taller blueberry bushes. But these always stay small like this. And right here, I see if I went to count these, I would see clumps. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 clumps right here. This is probably all one plant. Probably all the same plant right here. So they grow underground, and you call that clonal reproduction. So they do have flowers and seeds. They're pollinated by insects. So they have sexual reproduction, but some plants also reproduce clonally or asexually. Now, not here, but I don't know where it was, but I was reading about these earlier, and they can make mats that are as big as like more than a half a mile in length, and can be over 1,000 years old. That wasn't here that we did that research, but wow, that's amazing. It doesn't actually surprise me that much because other plants in the scrub are also super old, like the palmettos here. Well, actually, that palmetto there is not super old. That one's a scrub palmetto, and they're younger. But there's some other palmetto behind me, and those can be thousands of years old too. And we have done that research at Archbold. Okay, cool. I'm just happy to show you. Spring is starting to be in the air here at Archbold. We're starting to see some blueberries flower. Okay, we're gonna bring AJ back on, and you can get a little peek inside her world here as a researcher at Archbold. So AJ, you want to stick your camera back on?
I absolutely can. Hello, how's everybody doing again?
I'll give you 5 more minutes.
Okay, so we're gonna do a quick tour. I'm gonna start by saying hello to our RA, Lexi, and our other intern, Haley. So I'm just gonna show an old intern project. I hope you guys can see it. It's a lichen project. I don't know much about it, but it's a different species of it. Really amazing. And then we're gonna go downstairs because I have some really cool tools for you guys to see. So what we do in the field is mostly just like maybe tagging plants, checking when we do demography, we're checking to see, the plants that we may have tagged like 3 years ago, are they still there? How tall are they? Have they like gotten any wider? Are they reproductive, actively reproductive? So I'm gonna show you guys, let me turn on my camera. All right, this right here is Lucky. This is our metal detector, which detects our tags and our flags so that if we ever lose a plant or we can't find one and we need to use our GPS, we can use Lucky to just scan the area and hopefully we catch something. These are our 2 different types of GPSs. This is the Garmin, a little bit smaller. And then this is the Trimble, super expensive, nice and heavy, but this can get you pretty much to anywhere. Yesterday we did Diceranda Christmanii, and this is like one of the maps from there, and this is just like a data sheet. These are the tags. We have so many tags because we have so many plants. And then sometimes our plants are really tiny and we can't use tags and we can't use flags. So we have to use little toothpicks, and they all have these different shapes. So we have spades, we have hearts, we have diamonds, and they're all in different colors just to identify them even more. And then one of my favorite parts of this is we have a herbarium here. So I'm going to show you guys how we mount our plants. After we use this really cool adhesive glue, we then, kind of like paint it on to these plants and then we mount them. So these mounts are like little paperweights, and we use them to hold the plant down so they stick a little bit better. And then when it's all done, it looks something like this, really pretty. And now I'm going to show you guys our huge herbarium. So many awesome plants. There you go. So just imagine every little one of these photos has maybe 1, maybe 2 plants in them. And it's 3 containers of them. So many of them that had to be hand pressed, hand dried, hand, glued, and then mounted, and then put into these in a certain order. To just give you some ideas of like what goes on in here. That's just a little bit of it. Oh, and one of my favorite parts of this, for obvious reasons, is this huge map of Archbold, which I'm going to end with because I don't know if you guys know this, but Archbold is pretty big. And I think that giving this perspective of it, of how really big it is and how many lanes we have and how many areas we have, it's really beautiful. So this is another view of our lab. And then it's back to me. So thanks, Dustin.
Oh, that's cool. Yeah, I love those maps. You know, when you're at Archbold and you go in different offices, different buildings, there are always maps and cool photos. Part of that is because we actually have a whole office that makes maps, map making office. I'm going to show one last thing here, and then we'll get to the Q&A, the question and answer with AJ. There was that question about trees. So let's just take a look at this for a minute here. This is that slash pine that I mentioned. And one thing that you notice about it right away is that the bark is very flaky, like paper. That and that it keeps its branches really high. You know, it doesn't look like a Christmas tree. There's not low branches here. And this is how it is adapted for fire. So plants and animals, depending on where they're located, they have to be built for those conditions there, for the soil, for the climate. Here, it's fire is a big process out here, and the fire can come through here, and it might burn up this flaky outside layer, but then that protects the inside of the tree, and it keeps those branches high so the fire never reaches the branches. A tree like this can last through many fires. It doesn't last forever, and sometimes a fire will get it. A lightning strike might get it too. And if it does die, it might actually just stay there for 80 years, 90 years without even falling over, which is pretty amazing. I did bring, this is a slice of one of these. And when you look at it, you can see all these little tree rings. When you're looking at tree rings, you'll notice there's lighter rings and darker rings. Both of those rings make up a year. So it has the summer growing season in there is the light area, and then the darker area is the winter where it grows slower. Now, in some places where you don't have seasons, like in the Costa Rican rainforests, the trees there do not have reliable rings because the climate is pretty similar throughout the year. There's just the growing all year long. So it doesn't have reliable rings. But I do have the macro lens, so we're pretty much out of time, but let's just take a peek at this with the macro lens. And you can see these beautiful details. You can see the two different bands making a year. I haven't counted all the rings on this one, but I don't know, maybe 60 years or something like that. Pretty cool. Sometimes you can even find diseases or damage from fire. There's a whole story in these. Oh, let me switch back here. It's actually a type of science called Dendrochronology, which is using tree rings to look into the past and learn about things like fire and climate. It's pretty cool. I hope you've been adding some questions to the Q&A. It's been fun. It's been hot. I've been sweating. If you're in a class and you need to get going to your next thing, then feel free to do that. It was wonderful to have you on here. And if you can stay on for another 10-15 minutes, we'll get to questions. So have a good day, friends. Hasta pronto, amigos. It's wonderful to have you. AJ, oh, and then also, yeah, remember to come back next Tuesday. AJ, do you want to get your video back on? And Margaret, do you have any questions for her?
We do. I'm going to start with one quick question for you, Dustin, because it has to do with the tree slice that you just pulled out. Someone wants to know if you know how many rings are on that.
I haven't counted that one, but just like eyeballing it, estimating, it looks like it's around 60 years old, somewhere in that range.
Awesome. Alrighty, we do have some questions for AJ. The first is, can you tell us more about what Zizyphus is?
Okay, yeah, I can. I probably couldn't tell you everything about Zizyphus, but I could tell you a little bit. Its scientific name is Zizyphus Salada. It has these really pretty flowers on it. They're really small. Not something like you would like notice. They, when I was out there in January, we had some flowering, but most of them were not. And they're like these yellow and white flowers, really tiny. I wish I had a picture of them. But yeah, they're, like I said, endemic to Florida. They primarily grow in yellow sands. They're really thorny. The whole month of January, my hands were like full of thorns, so they fight back. And yeah, they have like multi-stems. Anything else you guys wanted to know about them? Like, I think that's, I'm thinking about more like visually what you guys would see, but you probably wouldn't see Zizyphus just like walking down the street. It grows in some weird places. We've been to like some really weird places, like really disturbed parts of farms. Like we would be out there in the middle of the day and then like a bunch of cows would just come and like watch us. It's not like in these random fields. So they grow in really weird places, but that's pretty much, I think, the extent of what I know about the Zizyphus plant.
A follow-up question is, do they make fruit?
Yes, but it's not like the fruit you would eat, but yes, they can fruit.
And I have a follow-up question for myself that I think people will probably be interested to know, which is, how often do you go off-site to do research or to take these measurements?
A lot. So the whole month of January, Zizyphus is not on this station. So we had to like haul to like maybe hour or two to get all across the Lake Wales Ridge for Zizyphus. But even for any other plant, like we're pretty much off the station at least once a week, if not more. So we're all up and down the Central Florida area.
And another Zizyphus question is, do they have any specific pollinators?
That's a really good question. I don't know the answer to that.
That's fine.
Specific pollinator. That's actually a really good question. I'm going to write that down because maybe it will hit me as I'm walking out. But as I know, no. Yeah, I'm going to say no for now. But give me like 5 minutes and I probably can find a better answer for you guys.
And we send out an email after all of our nature walks with the answers to questions we might not have known the answer to. So AJ can also check that and she can let us know and we will email you the answer.
Absolutely.
As well.
That was a really good question.
And so we have some herbarium questions as well. A few of them falling along the lines of, do other researchers come to look at your herbarium, or have there been other research projects that people have used that maybe aren't Archbold scientists?
So since I've been here, of course, like I haven't seen any researchers come because of COVID. So I'm pretty sure they would before all of this, but do I know that specifically? Probably not. But they people do have special projects. Like there's a lichen project going on. One of our RAs are like, I think repressing or looking at certain plants in one of our fields. So there's projects like Carter Creek, but yeah. So there's
I can add to that one because I've been here 8 years. So definitely, yeah. We have our own Archbold people that are on the, getting paid by Archbold. They're part of our staff. But then there's a lot of visiting researchers from different universities that come here. Some of them come year after year after year, and have a project that might last for 30, 40 years, which is pretty amazing. But other times it's like a graduate student or somebody that's earning their PhD and they need a site to do field work at. So they might be at the University of Florida and come to Archbold for a month or a couple months a year, that type of thing, while they're working on projects. So we do have all kinds of different collaborators, and a lot of our collection is also online. So researchers today, whether it's the insects or the plants, can go online and see specimens from herbariums from all over the country, and see where people were finding certain things. It's pretty cool.
Yeah, I can add to that too. That each plant at the bottom of it has a special ID. So like when you scan it, it takes you straight to like online to see the plant. So yes, everybody has access to our herbarium.
Alrighty. I think that's all the questions I see for AJ right now. Feel free to send them as well. And she can also answer questions just about what it's like to be intern here in general. I will say something that she didn't mention is that as well as being interns here, both of us actually live on site. So that's a really fun thing too, is that we get to study in this area and we also get to live here, which is really, really fun and a really unique opportunity. Back to some tree ring questions that I'm seeing. The first one is the rings on the slice are not concentric. It seems that the center is way off. Dustin, do you know anything about that?
Yeah, every one of these is different. And if you, if you really start taking a look at trees, or if you watch a tree grow over years, they're responding to what's around them. So maybe there's more light on one side and it's shadier on the other side. Maybe there's more water on one side. It's next to a creek or something. So there's all kinds of different reasons that would make the plant grow differently. Remember, these are living things. They're not machines. They don't grow perfectly symmetrical. Every year they're laying on another layer, but it can be thicker on one side or another. Let's say the tree is starting to lean a little bit one way. Well, maybe it'll start growing thicker on that side of the trunk to support itself so it doesn't fall over. Or it has to heal from an injury. So they, just like us, they are, each of them is unique. One of the cool things of looking at them, they're all different, all a little different, and have their own story.
Awesome. Okay, I lied. There are more questions for AJ. First question is, are you trying to catalog every species that lives there, that lives here?
I think that's the goal of every herbarium. So yes, but as of me actively trying to get every species, no. But yes, I would, that would be the goal, the most beautiful goal. But is that possible? We'll see in the next couple of years.
And for the herbariums, does it allow you to track plant phenology as well? As in, do you collect different stages of a species?
I have not. I've only done like a couple of days of herbarium, but I have not. But I would imagine that they would, like if it was like a plant that was something that, I guess from like when it was first like sprouting to like it being a full adult, that was something that was completely different. And I guess for herbarium uses, people really needed to know like these are the same plant, but just they look completely different in different stages of their life. I would imagine that they would do that. But as for me, I have never done that, different phenologies.
And
so I can add a little bit there. Some of the plants here, back in the day, people thought, this is one plant. And then over time, there's enough evidence gathered to actually show these are two different species. So having those herbarium specimens is helpful.
Mm-hmm.
And I'm thinking particularly of some of the scrub species. So some of the oaks. So right behind me, this is Myrtle Oak, this bush here. And there's another one that looks a lot like it called Fucusina pina, or Archbold oak, or scrub oak. And that was one that in the 80s, they were able to show there was enough differences between how they looked, as well as, I think, I don't know all of the ways they used to determine it, but then they could show, hey, we actually do have enough evidence to show these are different species. But sometimes one plant species can have a lot of variety, and the same species, you look at a leaf from one tree and another tree, and they can look a lot different. So it is good to have lots of specimens. We don't just collect one, we're collecting them throughout the years.
Another question for AJ is, someone wants to clarify, did you say that you were collecting seeds from some of the species? Is that for restoration efforts? Are you trying to get any of them to grow in more places?
You can collect seeds, yes. But I guess for the example that I put on there, we were doing clippings to give to Bach Tower because we were going to do like an outplanting type of thing. So like reintroduce it to somewhere else. So yeah, but you can, that's absolutely possible.
And for those that don't know, Bach Tower is I believe it's officially a botanical garden potentially, but it's also in the Lake Wales Ridge in Florida. And so there's some exchange between our two organizations. Another question is, do you know how far back the herbarium samples go at Archbold? And that the old samples seem like they'd be really cool.
Wow, I do not. Now that I'm asking that. Lexi, do you know how long this herbarium has been going on?
The oldest one I have seen is a collection from 1977, and that's the oldest one I've seen, but I think there were older ones.
Okay, so Lexi RRA has been here way longer than me. And she said 1977 is the oldest one that she has seen, but she's pretty sure that there's more that happens that have been before that.
And I don't know either, but I could say Richard Archbold he was collecting specimens back in the 1930s before he even visited here, but his 1930 specimens pretty much all ended up at the in New York City at the Natural History Museum up there, because he was connected to them. So my guess is that some of those early ones from Archbold probably also ended up up there in New York.
Another question, which could be for either of you, which is, can specimens change over time in a species? And do you think this could help determine climate change effects?
That's a really good question.
Yeah, I can take a crack at that. Having long-term specimens, whether it's insects or plants or whatever, we have collections of all kinds of stuff here. It is really important to see how things change. And I don't know at Archbold specifically of seeing anything of the researchers knowing of plants looking different or animals looking different here, but definitely around the world, research has shown, for instance, the color of an animal that is, say, in a city has been selected over years to blend in better in a city, or maybe in a new one example from near here would be the bird, the Everglades snail kite, which has grown longer, bigger beak to eat the invasive snails that are here. A new food source, hasn't been here that long. And the researchers showed that within just 10 years, the bigger birds were surviving more and having more kids. And so they were adapting, evolving right in front of our eyes to this new food source. So having specimens can definitely help with that.
Alrighty, so a few more questions, but I will just point out something that came to my mind was that, so something that's really interesting for AJ and I is that internships typically last here between 6 to 10 months, which is really great because it gets a lot of researchers involved in really, really long-term projects, but it also kind of puts us in this dynamic where we see a very, very short thing of like this incredible project that's been going on for 50 plus years. And so we might focus on something really, really specific. And then in 3 years, they're focusing on something else super specific, but it's all technically the same project, which is really awesome. It's incredible. It's wonderful exposure, but these questions can be a little tricky for us. Another question going back to the tree rings that was talked about earlier is that if there are certain rings that are thicker than others, does it mean that that tree grew more that year? And does the difference in rings give you more information about the height or the diameter of growth each year? Dustin?
Oh, good questions. And I'm not a dendrochronologist myself. Every species is different. So that's one thing that some species are more reliable in their rings than others. Sometimes you can have fake rings that for whatever reason it added an extra ring that year. But yeah, you can learn a lot about it. I don't know for the slash pine in particular if you can tell how tall it is based on the diameter. When it comes to ecology and helping to protect some of the rare species here, I know that we work with a bird called the Red-Cockaded Woodpecker, and it's an endangered species, and it lives in sometimes it's in slash pines, but generally it lives in longleaf pines that are still alive. And in order for the biologists to determine which ones would be good homes for them, they measure the diameter. They don't look at tree rings, but they measure the diameter of the tree. And so how old a tree is, how thick a tree is, it's interesting to us from just the curiosity part, but it can actually be a part of keeping a species from going extinct too. So with the case with the longleaf pines, this area, almost all of Florida, or a lot of Florida, was logged in the early 1900s. So the pine forests, there's only 3% left of the original pine forest. And now those trees are younger, so they're skinnier, they're smaller. And what you need generally is a tree that's over 100 years old for an RCW, that woodpecker, to make its home in it. So if you have younger trees, the biologists actually need to climb up them, use a chainsaw and cut a hole in them, put a birdhouse inside of it to make it easier for the birds. But they still want to find the biggest trees they can.
Alrighty, I'm just gonna ask a few more questions because we're running out of time, but we do have a ton more, so we will be answering those in our email afterwards. The first is from Rena on Facebook. Do we know what the oldest age of a tree recorded on Archbold is?
Ooh, that's a really good question.
Well, I would say that for tree rings, I'm not sure. None of the trees here get that old above ground because we have fire and lightning. So maybe you've got a tree that's 100 years or something. But the cool thing is, remember I mentioned earlier that we have clonal plants like some of the palmettos here. So they're growing underground, and the palmettos, the clumps of them, we have measured their age. And the average age is around 5,000 years old. The oldest was around 8,000 years old. And that was a researcher that's one of those visiting researchers that comes down every year, Warren Abrahamson. Mm-hmm. You might notice that Margaret's intern name is named the Jill Abrahamson Memorial Internship, which they sponsor. And he's been coming down here for like 40 years studying palmettos and other plants, which is amazing. So we do have some super old plants here.
All right, I'm gonna go ahead and stop our questions there, just because we have so many more. So we'll have to answer them in our afterwards email, but thank you to everyone who hung on till the very end. A lot of you did, so that was awesome. Dustin, AJ, do you have anything else to say?
No, thank you guys for coming out. We appreciate it. This was really great. You guys had some really good questions. I was kind of stumped on some of them, but hopefully we can answer them in the email afterwards.
Yeah, and I think it's cool to see that just because somebody works out here doesn't mean we know the answers to everything. Sometimes people expect us really to be just like walking encyclopedias. I will say some of the older biologists that have been here for 40 years are pretty much walking encyclopedias. But it shows that you can still be a professional and be a scientist, and you don't know every single thing. So don't be worried if you feel like you're not smart enough or you don't know everything. You can still be a scientist.
Yeah.
And I just want to say thanks, AJ, for coming on. Maybe we'll get you on another one of these too.
I would love to. This was so much fun. Thank you guys.
Alrighty, thanks
Bye,
everybody. We will
Bye,
hopefully
everybody.
see some of you all next week when we do the same thing at 9:30. Bye everyone, have a good week.